So I would like to welcome you to 212, Bioengineering 212, a class on systems biology. I'm Bernard Paulson. I'll be delivering these lectures. They will be recorded, so we encourage you to postpone questions until afterwards. The lectures come from the book pictured in this slide, and each lecture given here will correspond to a particular chapter in this book. So we are going to start out with a lecture on the introduction to systems biology, where I'd like to talk about this field broadly, where it came from, what it's all about, what are the core values in it, what are the paradigms that are developing, and so forth. So the outline is as follows. We'll first talk about the grand scheme of things, and this will be focused on the genotype-phenotype relationship that is so foundational to all life sciences. We will then talk a little bit about the basic features of genome-scale science. As you know, in the mid-1990s, the first genome sequences came out, and we've been mining them now for 20 years. And because we can now take genome scale points of views of organisms, we have something that's called genome scale science. And the type of systems biology we're going to go through here is an example of that. We'll talk in particular about the systems biology paradigm. Every field is built on certain paradigms, and it's important to articulate them and understand them, and so we will do that. And at the end of this lecture, I'll just make a few comments about the foundations that are being built for this field. It's becoming fundamental in nature, just like PChem, if you go take a class in PChem, or in other fields. And so we'll summarize these at the end. Okay, so we'll start then with the grand scheme of things. Think about life sciences broadly and where systems biology fits into the life sciences. And since we want to take a broad point of view, I'm going to start talking about this gentleman here that all of you know, Gregor Mendel, who studied breeding of plants and was mixing plants together and studying the progeny of such mixes, and came up with the basic laws of genetics. Looking at it now, retrospectively, you would say that Mendel discovered the existence of discrete quanta of information that travel from generation to generation, and these quanta of information affect the form and function of the organism that we recognize as a phenotype. So these quanta, and then there are the phenotypic consequences of that. A little later, this became more formalized through the work of a Danish gentleman called William Johnson, who defined the term gene, talked about the genotype, certain set of genes, and the phenotypes that result from that. And this kind of led to the formalization of the notion of a genotype-phenotype relationship that is so fundamental to biology. So we all have our genes, all these quantum information, that create form and functions of an organism. Now, these quanta are not immutable. They actually change. So we can have the same quanta of information in us, but they may be slightly different, and we'll, of course, talk about that later in the class. So genetic variation, in other words. So there are some well-known cases of the exploration of this relationship, and many of you saw this example in some of your earlier textbooks. This was Linus Pauling's work on the mutation in beta-globin and hemoglobin, where this one mutation in this one gene was manifested in a phenotype, sickle cell anemia, that is a pretty severe disease. So this is an example of one gene, one mutation, and a phenotype. But there's also interesting biology in it, because this detrimental mutation survived under a selection pressure provided by malaria, because the malarial pathogen infects red cells and has to incubate in them. And if the red cell had a shortened lifetime, that life cycle couldn't be completed. So it's kind of interesting example from a number of standpoints. Now, there are many such monogenic traits that have since then been described in the human population. And you can neonatally assay for, I think, 150 or 200 of them. Not all of them are as serious as sickle cell anemia, but these are various traits of human beings that are associated with a single gene, which is interesting. So these are called Mendelian traits because there's one gene that, and alterations in one gene that lead to a phenotype. But for most other biological functions, it is not that simple because you have many, many gene products come together to execute a biological function. And they all contribute to the phenotype, maybe in different ways, so potentially mutations or changes in any of them can affect that phenotype. And to get at that, we of course need systems analysis. We need to identify these, all these genes that participate in a particular trait, assemble their relationships, model them, and then try to describe the systemic nature of that relationship. And this is, of course, a big part of this class. So this was in the 50s. This was conceptual, well, actual for this case, but broadly speaking, the genotype-phenotype relationship was conceptual. Now, in the mid-90s, that started to change. So Craig Venter, that I think most of you know or know of, he sequenced the first genome in the mid-1990s. And we started getting genome maps like the one shown in this slide that we are all now very familiar with. This was extraordinarily important because once you have a full genomic sequence, you can in principle define all the genetic elements that make up an organism. They should all be on that genome. So now we could, I should say, for the first time, define the totality of the genetic makeup of an organism. Now the reality is that we still don't understand what all the genetic elements on a genome do, but for simple organisms, we have a pretty comprehensive knowledge of what the gene portfolio is and what their individual functions are. So this was a significant development because of the scale. Now we could do genome scale science, and in principle, look at all these genes, all these individual quanta of information, and try to trace them through to biological functions or phenotypes. That process has really been unfolding over the last 20 years. So with the genomes and their annotation came this challenge of putting the pieces together. And there were two gentlemen that were actually graduate students here in bioengineering, Christoph Schilling and Jeremy Edwards, that started that process. They began to look at well-characterized model organisms or those with very small genomes. So H. pylori, that infects about half of mankind, has a genome of only 1.7 megs, or something like that. And they would rummage through the annotations of these genomes, pick out genes with known function, define their biochemical functionalities, and started piecing together networks based on that. And the easiest set of gene products to deal with, at least then, were the metabolic genes, because we know so much about metabolism and the enzymes that carry out basic metabolic functions. So the result of such a reconstruction can take on many forms. One of them is a genome scale map of a metabolic network, and that's what's shown there. This does not look that different from a picture in your biochemistry textbook, in Lehninger or Stryer, or whichever textbook you actually read. But the big difference here is that every single one of these transformations, these chemical reactions, is tied to a particular gene in a particular genomic location in that organism. So there's a full and explicit genetic basis for that map. And that opens up all sorts of possibilities that we will discuss in this class. So that's very important, that this is a genome-specific map representing a particular organism. So this became possible in the late 90s for bacteria. This graduated to eukaryotic cells. Here is the first genome scale map for metabolism in a eukaryote. The yeast we all love, Saccharomyces cerevisiae. Yeast generate beer, wine, cheese, and other good things for us. Important industrial organism. So this was the first eukaryotic metabolic map that was put together. And a few years after that, the human genome became more and more complete. And by the fall of 2004, Build 35 of the human genome was published, which covered 99.99% of the euchromatin where the genes lie. And there were a number of online resources appearing at that time that had information about the annotation to these genes. And you may remember, but maybe you're not old enough, but 10 years ago, there was this substantial downgrade of us as a species in terms of the number of genes that are on the human genome. First, people thought there were 170,000, then 100,000, then 70, but in the end, it's somewhere between, 30,000 plus minus 5,000 genes or something like that. So this is from that era when we started realizing that was the case. So this reconstruction here of human metabolism, called RECON-1, was put together by 7 people that are shown in this slide. Students in bioinformatics, students in bioengineering, students in the medical school. And this is important to emphasize because a reconstruction is a lot of work. And in the end, it's fundamental. Everybody wants it. Everybody wants to have it as accurate as possible. Everybody wants to use it. So there has to be buy-in from the community that the reconstruction is accurate as we possibly can make it. So we'll talk a little bit more about that later, the actual workflows that underlie a reconstruction like this. I should emphasize that this is the global set of metabolic capabilities on the human genome, and they are not all expressed in every tissue. Every cell type in the body or the tissue type in the body uses this network in a different way. So we have to contextualize this network when we want to study a particular cell or a disease process. And that's normally done based on expression profiling and other methods we have to determine what part of that map is actually active in a given cell under a given set of conditions. So these are some examples. I could have picked others, but these are kind of important organisms and milestones in this history. We can now generate reconstructions for metabolic networks, and we'll talk about that here. We can now generate networks for protein synthesis. We can generate networks now for transcriptional regulation. We now have workflows that demand the input of a certain data types, and if we have them, we can reconstruct those networks. So even if metabolism was the first case where this was realized, we can now do this for other networks, and a lot of the mathematics that they used to analyze them were developed around metabolic functions. And as many of you have heard in the popular press, metabolism is percolating to the top in all the major categories of human disease. Cancer maybe be the most familiar one lately, but even psychiatric and neurodegenerative disorders have a strong metabolic basis. So it's important to have these networks. What is shown in this slide is rather important. It's subtle in the beginning, but it's very important, and you will appreciate that importance as this class unfolds. What a network reconstruction is, it's a knowledge base. It's not a database. It's a knowledge base. It's conclusions, it's statements that are derived from a lot of different data types. Hexokinase, phosphorylates glucose, and here's the reaction. That's knowledge. Here is the gene that encodes that enzyme, and here is the amino acid sequence. Here is the genomic location. Here is the promoter to that gene. Here are its properties. This is all what we consider to be knowledge. This is basic knowledge about biochemistry, molecular biology, and genomics. What the reconstruction is the representation of all that knowledge on a genome scale as well as we know it for a particular organism. So reconstruction is a knowledge base, it's not a database. And we call it a big database, the 2 Gs, because they are biochemically, genetically, and genomically structured. And we will undoubtedly see protein structural information and so forth, find their way into these knowledge bases. So that's the first consequence of this effort. We have now knowledge bases. And what's extremely important is that we can now take all that knowledge, what we consider to be knowledge, and we can code it up mathematically. And one example of that is shown on this slide over here, which is a table of stoichiometric coefficients. And I'll talk much more about this later in this lecture and throughout the class. It's an assembly of all the stoichiometric coefficients that represent those chemical transformations that we have assembled. So this was really the birth of genome scale biology, systems biology, because we had genome scale representation of all these genes and all their functions, and we could now reconcile their integrated functions through a mathematical model. And therefore we could compute functional states of networks, which is the actual biological functions that they carry out. So this kind of completes formally the genotype-phenotype relationship. You've heard people talk about this relationship, wave their hands and describe it in qualitative terms, but now for the first time we had equations. We could actually calculate things, and they were all based on mechanisms, genome scale mechanisms of enzyme actions, gene functions, expression, transporters, and so forth. And this is a fundamental transformation, or I would say a fundamental event in the history of life sciences. We finally have fundamental equations and laws and principles like, say, physics developed 100 years ago or so. And this is a process that I think is just starting. I'll introduce you later to the notion of dual causality later on. In chemistry and physics, we have what's called proximal causation. You do something and there's an immediate reaction. In biology, we also have what's called distal causation, because with subsequent generation, the properties of organisms change. They adapt, they evolve. So the way they respond a few hundred generations later, say, on microorganism, is different than maybe the organism did that you started with, because the genetic makeup has changed. So there is distal and proximal causation that has to be described in these models, and that makes modeling in biology fundamentally different than in physics and chemistry. As we will see, we describe distal causation by an objective function, which is the function that represents the selection pressure and the desired functions of the organism, and that becomes the biologist's function. It doesn't exist in chemistry, it doesn't exist in physics. The proximal causation, on the other hand, we describe by the basic physical laws and data and some other inputs that we will talk about. So that's the grand scheme of things, where we have come from since Mendel was breeding his plants. Now we have genome scale models of organism functions. Now with this development comes a large number of conceptual developments, many of which we have to be familiar with, understand how to deal with, think about, and so forth in this field. And I'd just like to go through a few of them here. There are many more, but there are a few concepts that I picked out and put into chapter 1, and I'd like to describe briefly now. So on the left hand of this diagram is what used to be the driving force in molecular biology, biochemistry, and that was to find components in cells, isolate them, and characterize them. You could maybe delete them from the genome of a cell if you were lucky, or get a genetic variant to try to ascertain the in vivo function. But by and large, most of this information was obtained ex vivo. You would rip a cell apart, isolate the component, and study it biochemically, publish some nice papers. So from that comes what we call the bibliome. So there's a lot of literature on all these experiments available, and that literature is key for us to understand the biochemical function of a gene product. A typical annotation, genome annotation, does not have that information in it. It'll just say this is lactate dehydrogenase. It won't tell you anything about thermodynamics. It won't tell you about the cofactor use. It may not tell you about the cellular location of the gene product. All of that information is found in that bibliome that was unraveled over 60, 70 years or so. Now, like I said earlier, in the mid-90s, we started sequencing whole genomes. Expression profiling arrays came out. People talked about, in optimistic terms, about proteomics and metabolomics at that time. Maybe 15 years later, that those visions are being realized. But they talked about this profiling approach to biology, genome scale measurements, genome scale assessment of different properties of organisms. Technologies that we think of now as under the name of omics data that results from those technologies. So when they started coming out, bioinformatics really came into being as a field. There was this incredible need to be able to manage all this information, because genome scale is a big scale. Not only to manage the information, but also to model it in a systemic way, based on all the rationale I've just gone through. So this is important, that life sciences have kind of shifted in this emphasis from looking at individual components to looking at cells and organisms as systems of molecules that come together to execute cellular functions. So here is another important concept. We're going to start to introduce the notion of modules. So a gene product by itself doesn't do much, but when you put it in the context of other gene products, you start to see their meaning. Like, for instance, for glycolysis to be active, you have to have 12 chemical transformations there. A signaling pathway has to have a receptor, some submembrane transactions, translocation into the nucleus, activation of gene expression. So it's like a whole pathway. So we think of modules like that in metabolism, and that's an old concept of a module. We also think of molecular machines, that many of these gene products aggregate in space and form super molecular aggregates, the ribosome being a very familiar one, DNA polymerase and so forth. So this is kind of the low level of thinking about how gene products come together to execute our functions. So we think about circuits, genetic circuits, and molecular machines. But if we think a little more broadly, we see that a pathway like this one, a biosynthetic pathway for an amino acid, actually is a part of a network. It's just a piece of that network. A pathway doesn't function independently of everything else inside the cell. So when we look at the functionalities of a pathway, mutations in it, gene knockout or whatever, it infects the intracellular environment that it functions within. It perturbs the whole network of interactions around it. And that's why a genome scale point of view is so important, to make that assessment as accurate as possible. Now, these very complicated systems operate in volumes on the order of a cubic micron. They're tiny. And there are literally thousands of different types of proteins in there, maybe thousands of different kinds of small molecules, metabolites, and so forth in these tiny little volumes. And if you start thinking about the biophysics here, what will it actually take to make that happen in a coherent way, proximity of the gene products, the concentrations, this and that, you start realizing that there are incredibly many constraints on this function. Molecular crowding of the volume, membrane area, turnover rates of an individual enzyme that determines how many of that copies of that enzyme you need to execute the function. And all of this has to be in balance. And quite frankly, when you start writing out all these constraints, it is surprising that a cell can actually satisfy them all, that there is a set of parameter values that makes this function coherently. Another way to state it's a restatement of a sentence or a statement that is often made, which says life is very improbable. It is rather improbable. There are relatively few parameter values that will make this function coherently. So all of this leads us to the view of cells in a hierarchical fashion. I mean, the genotype-phenotype relationship is hierarchical. It goes from the individual molecules all the way to whole cell functions. And if we think about the history of science, when you think about chemical causation or physical causation, we may think of people like Boltzmann and the Boltzmann equation and so forth. think about thermodynamics, kinetics, thermal motion, and stuff like that. Very much causation in physical chemical terms. But at the other end of the spectrum, we think about biological causation. We think about selection. We think about willful generation of alternatives. We think about mutations. We think about very different kind of causation at that level of that hierarchy. These, of course, are familiar, have been familiar. And systems biology has developed as a field to try to bridge this gap. And you see this this layering, this multi-level relationship that develops when you start looking at that. You may think of genetic circuits or molecular machines. You may think of networks and so forth. So a lot of systems biology is focused on trying to elucidate this understanding of the genotype-phenotype relationships. There are some shortcuts to go from genotype to a phenotype, but we'd like to fill in all the details in between. And there is that statement to Paul Nurse that is maybe hard to read there that says that one layer in biology will be informative about another and vice versa. So we talk often about coupling constraints. How do we couple these layers together to describe them in a coherent fashion? Okay, so these are some concepts in genome scale science and systems biology in particular. So let's talk a little bit about the systems biology paradigm that has evolved as a result of this and how we build curricula around that. So the systems biology paradigm is understood in terms of these four steps that are shown in this slide. First is this enumeration of components, what they are, whole genome sequencing, expression profiling of the multiple different conditions, and so forth, all this molecular profiling. Then there is that reconstruction step that I mentioned, is to build that network that underlies the function of a cell. And it's very important that in the first layer there are many bubbles, many technologies, but in the second layer there's one bubble. Because in principle, there's only one metabolic network in Saccharomyces cerevisiae, a particular strain. And it's, in principle, knowable. You can know what that is if you have sufficient amount of information. Then converting this into a mathematical format is a little difficult conceptual transition for many, especially those that have only been in life sciences. But many of you have done computer science, engineering, statistics maybe, and so forth. And this is not unfamiliar, I think, to most people in this room. And that is to take chemical objects, genetic objects, and convert them into a mathematical format. So once you have done that, and you've taken this reconstruction and described it mathematically, there is a number, but a discrete number, of methods to analyze the properties of a reconstruction. And some of them are indicated in there. I mentioned topology on one side. And maybe all the way on the other side, you might be wanting to study the stochastic nature of thermal noise in a network. So there are many different mathematical methods that can be deployed once you have the reconstruction done. And which one you use depends on what you want to know. And then we have finally the fourth step there, which is the utility of a validated model. We have high throughput data analysis. We have discovery of missing parts, something called gap filling. We can now systematize hypothesis generation based on data. We'll talk about this at the end of the class. We can now understand how biological processes unfold, adaptation to a new environment, oncogenesis, progression of any pathology, as we can see a time progression of changes in a biological system over time. And then, of course, finally, there's the engineering angle of designing biological functions. And we think of this broadly as synthetic biology. Very hot field, people talk about that quite a bit. How do I synthesize new biological function? The field in which that has developed the most is called metabolic engineering, where you can actually engineer metabolic phenotypes in production organisms to make biofuels, commodity chemicals, drug molecules, nutraceuticals, and so forth. And that's actually a pretty well-developed field, as it turns out. And there are many other kind of synthetic biology applications coming online. So how have we taught this here at UCSD? So there is a class that precedes this one, 211, that teaches these high throughput methods, the data types you can generate, and so forth. A little bit of reconstruction often covered in that class, but most of the network reconstruction material is covered in this class. Then there are the mathematical methods. The ones we teach in this class are more topology-oriented, constraint-based methods, studying stable homeostatic states and so forth. We used to teach in 213 how to deal with dynamic states of networks. That class has not been taught now for, I think, 4 or 5 years. But hopefully will be soon again. And then, of course, we go through the applications, using high throughput data, like I said, tailoring Recon 1 to a particular tissue type, seeing a cell that has been exposed to a drug and not exposed to a drug, profiling both of them, mapping it onto a network, and getting a genome scale assessment of what the drug perturbation actually does. And that's proving to be very surprising because since I'm on tape, I won't say much, but people think they know the primary target of a drug, but often that turns out not to be the case, or often, sometimes. We teach gap filling methods, algorithms like Smiley. So if there's a mismatch between prediction and data, we can now calculate what is the most likely reason for that discrepancy and do an experiment. So we can systematize basically hypothesis generation. We study, like I said, time-dependent phenomena, adaptation, disease progression, maybe the differentiation of a hematopoietic precursor cell to a lymphocyte. That's a well-defined process that can be studied now through systems biology methods. And then, of course, the designs. So we'll go through all of these topics here at the bottom of the slide in this course. So now, let me see if you had any previous inclinations towards systems biology. So you've all had biochemistry, right? Everybody here has had a biochemistry class. Okay, most, maybe not all, but essentially everybody. So you take a biochemistry class and you come across a molecule in a chapter like glutamine, and you're told this is an amino acid. And you learn about this as an amino acid, what it takes to place it in a protein and so forth. Then a few chapters later, you read about the urea cycle, how the body gets rid of excess nitrogen. Well, there is glutamine. It's in that urea cycle. And then you read a few more chapters and you see it's a neurotransmitter. GABA and glutamic acid are extremely important neurotransmitters. And you will see more functions of this molecule. And when you read each one of those chapters and you looked at the homework questions at the end of the chapter, they only pertain to that particular function of the molecule taught in that chapter. But as these chapters add up, you're probably thinking, just a minute, 3 chapters ago I was told glutamine does this, now I'm being told it does this. But glutamine does all these things. And many other molecules do all these things, like cyclic AMP, ATP, and so forth. They function in multiple different cellular processes. So it is impossible to reconcile those functions conceptually. And fortunately now we can do that mathematically. So if you ever had thoughts like this, you were thinking like a systems biologist, not about a single function of a molecule, but the totality of the functions that it can have, and not only it, but all the other molecules around it. And this is the magic of systems biology and the methods that have now been developed. We can actually do this, but you have to do it mathematically. So what underlies that process is the network reconstruction that I'm going to foreshadow here just a little bit. We are all familiar with a list of gene functions on an annotated genome, which is a list. If you think like a computer scientist, it's a one-dimensional object. It's just a list of names. Could be ordered based on the genomic location, but it's a one-dimensional object. Now, if you layer on top of that the functions of these molecules, you can see that the gene products are interacting. And that's shown in this slide. For instance, that heterodimer at the bottom of the slide, 2 of these molecules come together. Once they are assembled, then all of a sudden they catalyze a reaction. Or like shown on the top of the slide, this could be a transcription factor that's made by one gene, but it goes and binds somewhere else and affects the expression of another gene. So if you assemble all of these chemical interactions or chemical transactions, you actually end up with a table, a 2-dimensional object, where you have the components as rows And also there are different states, bound and unbound, for instance. And then every column in that table represents a particular chemical interactions between these components. And this is what reconstruction fundamentally is. It's a two-dimensional object. And with that table, there is actually additional annotated information that we can talk about. And eventually this, I believe, will become the basis for the biophysical manifestation of how a cell is organized and so forth. But that's yet to be developed. Now, with that comes another concept, another activity that we need to be familiar with. That's the notion of a workflow. To assemble all that information on a genome scale is a gigantic task. Error-prone, can be exhausted at night reading something, try to extract some information from a reference and so forth. One person doesn't do it. Normally, you have many people do it, often domain experts on particular gene products, downloading knowledge about them. And no one computational method solves all your problems. No one data type solves all your problems. So to achieve a good quality reconstruction, you have to go through a workflow. And that one is, a workflow is illustrated in this slide. The workflow that is now used to build a metabolic reconstruction is comprised of 96 separate steps that you have to go through. And there's an SOP that tells you how you go through all of these steps. Some of them are automatable, some of them are not. But there's a workflow that has to be executed with certain quality controls on it to make sure that the end product is accurate. So this is very important, a workflow. And those of you that are taking bioinformatics are probably excited about this because there are a lot of workflows in computer science. But in this, the workflow, in this field, the workflow also includes data types that get used. So this is one workflow. There are many workflows. But you know, like with a new experimental method or something that you put together that works, once you have a workflow, you can then apply it over and over and over again. And this workflow has now been applied to metabolic reconstructions around the phylogenetic tree. I think there's close to 100 well-curated, or reasonably well-curated metabolic reconstructions available right now. Personally, I think this field has a grand challenge, which is to go through that entire phylogenetic tree with a reasonable coverage of branches so that you can interpolate kind of between them, but get enough model organisms fully reconstructed so that we can understand the totality of all biochemical transformations that are currently resident on this planet. And I think this will happen. This will probably be one of these large science or big science projects. Will probably cost $100 million and take 10 years or something like that. But that's what you can do once you have a workflow. If you have a workflow and all the inputs are to that workflow are satisfied, you will get that output. Okay. So let's talk a little bit more about systems biology as a field, where it fits into, well, the biological hierarchy, if you will, so, which is shown here from a molecule to a population. So systems analysis in the life sciences has been practiced for a long time. Nobel Prizes have been awarded in immunology for systems analysis in immunology. There are models in ecology that describe by ecosystems. Physiology, ultimately, human physiology is kind of like a system. Systems biology, as we understand that term, is really molecular systems biology. It resulted from basically having the omics technologies, that we can profile all the molecules inside the cell of different types, whether it was the nucleic acids, the protein, the metabolites, lipids, and so forth. But somehow that word molecular got dropped, and it's just called systems biology. And understandably, it initially irked people in these other fields that you said, well, we've been doing systems biology for a long time, what are you talking about? What are we talking about are the molecules. We're talking about basically cell and molecular biology. All right, so let me then pick up what is, I think, the last topic in here, which is building foundations. This process of reconstructing networks and building models has proven to be successful. There are recent reviews that we will go through, or some cases that show that there is a certain range of biological functions that we can now predict. Not everything, we can't do everything in biology with these models. There's a range of functions that we can now predict and reduce to practice for a number of purposes that we'll go through. So, like every field, we need to have axioms. What are the fundamental statements? What are the fundamental truths that we build this field on? And they are actually pretty simple. There are 6 of them. Individually, they're all acceptable. But taken together, they kind of create this magic of systems biology. So the first statement is very simple, that all cellular functions are based on chemistry. So the Nobel Prizes are given in a certain order based on how fundamental the field is. So the physics prize is given first, because chemistry is derivable from physics. Then the chemistry prize is given, and then the physiology prize is given, because physiology should be derivable from chemistry. So underlying all biological functions is chemistry. So this is very important, that all models that we build, all reconstructions that we build, need to be chemically accurate. Otherwise, the foundations, the bottom level in that hierarchy, is incorrectly built. So this is very important. Axiom number 2, which is kind of new, which basically says that workflow that I showed works. You can execute the workflow, and you can use bibliomic data and annotated sequence data to build a reconstruction that accurately represents what an organism does. Now, cells function in a context-specific manner. This is very important. Cells have a homeostatic state tuned into them. So if you put them into a certain environment, they will execute a certain function. And therefore, we have to expression profile them maybe under that condition, or metabolomically profile them, or look at the inputs and outputs to be able to study that state. And that homeostatic state is a remarkably strong property of cells. Many cells that live in the environment only have really de facto 2 of those states, plus minus oxygen or no oxygen, for instance. And that's why they're so hard to cultivate, because we don't know what environment to put them in to make them grow. Other cells that we study in here, like E. coli, is a remarkably adaptable organism and multifunctional. It's easy to grow, easy to do this and that with it. But cells function in a context-specific manner. It's not just one function, it's context-specific. So whenever you study things in systems biology, the context matters. Then there is a statement that I made earlier, that cells operate under a series of constraints. For instance, these plants out there, they have to deal with the intensity of incoming photons. That's a constraint they have to live under. There's a certain amount of nitrogen and phosphate in the soil. That's a constraint they have to live under. I talked about the crowding constraints before. There's only so much you can pack into a volume. So there's myriad constraints on the function of a cell. The ones we can actually describe mathematically, we can derive directly from basic physical-chemical principles. And we focus on those in this class because they are fundamental. That's the first thing you got to do. Then you can map onto that environmental constraints, or you can map onto that regulatory constraints, which are self-imposed constraints by a cell, or you can map on that data constraints from that condition. But there are certain non-negotiable physical-chemical constraints that cells have to operate under, and those are actionable when it comes to building models. And then there is maybe the last axiom that some people may object to on religious grounds, but I think most people now accept them. I think even the Vatican has accepted the existence of evolution. But that is that cells change over time. They change with subsequent generations. And this is the distal causation that I was talking about. And it's extremely important that models are able to describe this. But there is a selection pressure in a given environment. Life is always creating different alternatives, and those are the most successful in terms of survival, maybe not the most fit, but they just survive in a given environment. Those are the ones that will continue. Look around this room. We are all different, but we are all outcomes of a selection process. So there are equivalent outcomes of a selection process. There are many outcomes of a selection process typically. But the selection process suggests that something is better than another. Because what is not good enough goes extinct, what is better survives. And that implicitly has built into a statement of some optimality principles. That there's something optimal about the function of a living system. How we describe that may not always be clear, but in some cases we can describe it mathematically. Mitochondria tries to make as much ATP as it can in a given condition. That's a statement that you can translate into mathematics and calculate with. If I evolve microorganisms in a nutritionally rich media, not environment, not nutritionally poor environment, you would assume that it tries to optimize growth, and that's indeed what it does experimentally. So we have objectives. We all have objectives in our lives. You have an objective to get a degree, for instance, because you're striving for that. This class may be a constraint. if it's a requirement to a degree, it's a constraint. You have to deal with it. But these are kind of the axioms that at least can be articulated at this point in time that underlie this field. And each one of them individually, like I said, can't argue with. But when they all come together, they generate the foundation for bottoms-up systems biology. Okay, so organization of the textbook we're going to use, it follows these basically 4 steps that are outlined earlier. We do network reconstruction here. That's the first part of the class. Then how we convert that into a mathematical representation. Then how we build models, predictive models based on that, and then how we use them. So we're going to go through all of these topics, roughly 2.5 weeks each for a total of 10 weeks. So the class will go fast. And it's a good idea to get focused on the term project sooner than later. And we'll talk about that probably more specifically next time. Okay, so we have a summary here. To summarize this introductory lecture, this background to systems biology. So first, genes are quanta of inherited information that travel from one generation to another, constantly scrambled up. In the case of eukaryotic genomes, constantly being mutated, they may be rearranged in the case of prokaryotic genomes, all sorts of other ways of, all sorts of other mechanisms active to change the genetic material. Some willful and some not, of course. So these quanta of information specify or lead to phenotypic consequences. We, 20 years ago or so, were able to sequence whole genomes and therefore enumerate all the genes in an organism. We have candidate functions for the majority of these, but not all, as we'll talk about later in the class. So having that list presented us with this grand challenge of trying to figure out how all these pieces come together now and create a coherent biological function. That was a grand challenge that kind of resulted from having genome sequences. And systems biology was a field that was developed in response to this grand challenge. So how do we deal with that? The interactions between these components are being documented. There are chemical transactions, there are physical transactions, and so forth. We are working these out. Based on this information, we can reconstruct these networks, we can validate them, we can test them, we can delete components of them, we can do all sorts of perturbations of them to try to figure out how good our reconstructions are. We can convert them into a mathematical format, which is important because now we can compute based on a knowledge base. And after some years, this basic paradigm developed that we have to enumerate components, either in an absolute or a context-specific manner. We reconstruct networks, we convert them into computational models, and then we study phenotypic states, both experimentally and computationally, and we're constantly comparing. One important consequence of this paradigm, which makes systems biology different than most other branches of the life sciences, is that it becomes inherently mathematical. We have to use mathematics. We have to use algorithms. We have to use computations. We have to use matrices. We use Boolean statements. We use maybe random fluctuations in concentration levels and so forth. So, the fact that we can now formulate these relationships and that our knowledge is complete enough to get good reconstructions, we can actually now predict biological functions. And this started happening maybe 3-4 years ago. And this is a major development in life sciences, is to be able to build a genome scale model accounting for all the gene functions you know to be present in an organism, and based on that predict phenotypic function. Responses to environmental stimuli, responses to single gene knockouts, double gene knockouts, and so forth. So this is fundamentally why this class is important, and hopefully you will enjoy it. So that's the end of the introductory lecture. Thank you for your attention.